So hello and welcome to the second lecture in the week 10 introduction to RNA-seq analysis. In this version of the lecture, I'll be giving you an introduction to an RNA-seq analysis workflow. This is one of a number of different workflows that are available for conducting RNA-seq analysis, but this is probably the most popular one in part because of its simplicity of implementation. So this is what we call the Tuxedo package. There are a number of softwares. If you've begun looking at the homework, you're aware of them. Bowtie is the aligner. So we've collected our RNA-seq data. This can be either for paired end or for single end RNA-seq reads. Bowtie is the software that does the alignment. This is a super fast aligner. Similar to BWA, which we used for the DNA sequencing. Bowtie 2 is the version of the aligner we're now using in this course. This is the newest release, and it is a gapped aligner similar to BWA, which means that reads can span small deletions. However, Bowtie cannot conduct what's called split read alignment. So while it can span short deletions, say deletions that are shorter than the length of the read, Bowtie cannot do split read alignment, which of course is necessary when we're aligning reads from RNA-seq. So imagine a read that spans an intron in a transcript. When you map this back to the reference genome, of course, the read is now split because of the intron, and so we need an aligner that can do this kind of split mapping. So this is what Top Hat does. So Top Hat actually is a software developed that will use Bowtie in its first sort of round phase of the alignment, where it does what's called exon first aligning, and then the second phase, it takes the reads that were not mapped by Bowtie, and then it realigns them by splitting them and looking for places in the genome where the split read both ends map. Top Hat generates a BAM alignment, which we then use various downstream tools to assess the various transcripts that are present in the alignment and independently derive read counts for each different transcript in the genome. And then finally, so this is the functionality of cuff links, cuff merge, and then cuff diff is the method that does the statistical analysis and gives us our p-values in a typical differential expression type of analysis. So that probably reminds me that I should say that this workflow is intended, for our purposes, for differential expression. So in the homework, what I have you do is looking at a rice data set in which we have a control sample of rice, a pair of samples, that is, where we have rice grown at a particular temperature, and then we have a treatment, a pair of samples where those pair of plants were grown at a much higher temperature. And so this is a sort of a conventional experimental design where we have multiple replicates. These are biological replicates, so separate plants that are being grown in each of two different conditions. And so in the analysis that you'll be conducting for your homework, there are a total of 4 RNA-seq FASTQs, which represent these 4 samples taken from each of 2 different experimental treatments. And the question that we're trying to answer is which genes in the genome are differentially expressed in these 2 different treatment conditions. So finally, CumRBun. This is the CumRBun package in R. We will be working with in week 11 to begin to summarize some of the results and generate publication quality figures. So let's look in a little bit more detail about what each of these different steps is doing. The bow tie is, again, our aligner that we use frequently in RNA-seq analysis. We could also use BWA, except remember that bow tie is implemented by Top Hat, where BWA is not. So bow tie is the first of the two phases of the Top Hat alignment protocol. So it's not a gap aligner, as I said. Bow tie 2 is. Bow tie 2 just has come out in the last, I guess it's a little more than a year old now. And again, bow tie does not split read by split reads, so it cannot map to reads that are at the junction between two exons in an RNA-seq data set. I should point out that, as with all of these tools, there's fairly good documentation online. And they're flexible. So there's a lot of command line options. Sometimes we are left as bioinformaticians with the choice of whether or not to try to come up with a perfect set of alignment conditions, or sort of using different options at the command line to try to come up with optimal alignments, or to proceed without trying to optimize all of those conditions. And sometimes there's arguments for proceeding each way. In any event, these tools are flexible, they're rich in their functionality, and you should explore the user manuals whenever you're beginning to work with a new tool. Top Hat, again, is sort of the alignment step in the RNA sequence analysis workflow that we're using. It runs bow tie for the first phase, and then it uses its own algorithm to split reads that did not map in the bow tie phase, and then try to use those reads to identify the junctions between exons and where they are in the reference genome. So, as we'll see later, both of these, the Top Hat tool can be run simply by specifying the reference genome annotation, and only use the annotations that are provided in the GFF3 or the GTF file that contains our genome annotation. However, for most applications, the authors of the Top Hat software and the workflow that we're working with generally discourage that. You may have read that in the Trapnell et al. Paper, which has been assigned for you and is available on the course site. This Trapnell 2012 paper, they basically argue that it's best to have Top Hat try to sort of naively infer the transcripts that are supported by the data, and not to rely on a already existing genome annotation. And the primary reason for that has to do with the fact that a lot of transcripts have yet to be discovered, even in humans and mouse, and some of the better annotations that are out there. Any one RNA-seq data set may in fact discover a number of new annotations. I'll come back to this issue in a moment. So Top Hat, in the second phase of the alignment, it splits the reads into 25 base pair chunks. These are the reads that did not map in the bow tie phase. And then it discovers these exons by looking for these coverage islands, these pileups, where the exons are. And then it uses a strategy here for dealing with reads, not unmapped reads, but reads that map to multiple places. And in the end, it weights those reads appropriately, and in the outputs, which are then considered appropriately in the statistical analysis downstream. So you don't have to worry too much here about the multiply mapping problem, simply because Top Hat has taken that into consideration. So in the end, what you get from Top Hat is an accepted hits.bam for each sample. So in the homework case, you'll be running this separately 4 times, once for each sample, and you'll get an accepted hits BAM file and an index of the splice junctions in a bed formatted file. So the next step is to run what we call cufflinks. So cufflinks is essentially a transcript assembly program that assembles the individual transcripts based upon the alignments that are provided by top hat and the index of splice junctions. So as you're aware, cufflinks, there could each of the genes in the genome rather has multiple different potential alternative splice variants, particularly in humans and other mammals, where any one gene has, on average, multiple different isoforms or alternative splice variants. Cufflinks is going to attempt to assemble all of the possible isoforms that are in the data that are supported by the criterion that it uses. So it estimates it calls transfrags. So these are not necessarily complete transcripts, simply because in a low coverage data set anyway, you may not end up obtaining reads that support the entire length of the transcript. So in cufflinks, they use the term transfrag to represent those potentially truncated transcripts. And then cufflinks, you'll see in the homework, its output actually does estimate the abundance of each of these different transfrags, but it in part uses those abundances to discard the immature primary transcripts. So as I mentioned in the previous lecture, there are transcripts that are detected at low level that contain the introns in them. Okay, remember, the introns are almost immediately spliced out, but because it's not instantaneous after transcription, and when we sequence very deeply, we start to pick these primary transcripts, the transcripts that have yet to be processed. And cufflinks uses the counts to detect the very low abundance ones and uses a method that we won't go into any more detail, but it uses a method to remove those transcripts that appear to have introns. So cufflinks you can also force to only consider the transcripts that are annotated, although in non-model organisms this is discouraged, and that's simply because there are likely to be many transcripts that are in the cell that have yet to be annotated. And then they relax this sort of best practices for humans, mouse, fruit flies, which are probably the best annotated genomes, and probably the majority of transcripts at this point, anyways, 3 or 4 years after doing RNA-seq heavily in these species, we probably finally have a fairly good sense of the full transcriptome under various conditions. So I guess the final point is here we run cufflinks separately again for each sample. So in your homework, you'll be running a cufflink separately for each of the 4 accepted BAMs that are generated from each of the 4 rice samples. So the next step is to run what we call cuff merge. Figure 3 in the Trabnell paper, which I'm showing you here in this slide, notice here how these fragments from each of the different replicates, so this is somewhat similar to our experimental design, where we have, there's 3 replicates in this experiment that they show you. We have 2 replicates, but otherwise we have condition A and condition B, which in our case is the rice control and temperature stressed conditions. And what cuff merge does is, so the goal is to identify, and quantify the abundance of each transcript. So what cuff merge does is it takes all of the samples and it identifies what it perceives to be, the same transcript, okay, across the different samples, and then builds a sort of a transcript model, which it then estimates the abundance of, okay, in each of the different samples. So you could imagine in a low coverage data set that you may not discover all of the individual exons, okay, in the samples from condition B, simply because of low coverage. Okay, so that's the problem. So what cuff merge does is it takes all of the data together, infers the model here, and then it estimates the abundance of this gene model or this transcript for further analysis. Okay, so actually CUF-DIF, I believe, actually does the FPKM calculations, but CUF-MERGE builds the gene models. Okay, so once again, as with all of these options I've mentioned so far, Top Hat and CUF-LINKS and CUF-MERGE all can use the GFF or GTF annotation as input, which assists CUF-MERGE in identifying transcripts. So finally, CUF-DIF is the tool that does the test for differential expression between the two conditions, that in our case is the temperature treatments in rice. It essentially will estimate the variance in the FPKM estimates across the replicates, and will then test the null hypothesis that the two treatment conditions actually are not different in their expression for each gene or for each transcript. So CUF-DIF will give you p-values for the gene models, each individual gene model, or you could just report a single p-value for an entire gene and ignore the sort of finer level of analysis of each individual isoform. So CUF-DIF does a number of things which we'll be exploring next week, including reporting the raw p-values and the p-values that are corrected for the false discovery rate. So this is kind of the recommended workflow. I should point out that in the Trapnell et al. Paper there are a number of modified workflows that can be adopted that you would use to in slightly different experimental designs. One possibility is to simply skip the CUF-LINKS and CUF-MERGE steps, so you can feed in directly the annotation to CUF-DIF plus the alignments produced by Top Hat and the splice junction predictions produced by Top Hat, and pass them directly into CUF-DIF. And in this case, what CUF-DIF will do is it will simply use the annotations produced by, use the annotations from the GFF3 or the GTF that you provide, and will simply use those gene models to obtain the estimates of the numbers of transcripts for each of the different transcripts in the genome. So CUF-LINKS, then, it's clear what it does is to assemble the different transcripts naively, using evidence from the data for individual transcripts, and effectively creates its own annotation of the genome based upon the transcripts that are in the data set. And then, CUF-DIF, if you go through the workflow we just went through, will estimate the abundance of each transcript and test for differences in expression for all inferred transcripts. So this is just one example of an alternate workflow. I will refer you to the Trapnell et al. 2012 Nature Protocols paper. That really is a paper worth going through carefully, particularly as you're considering other potential experimental designs and ways of analyzing the data. So this is just an introduction then to the workflow. We'll begin continuing our analysis of this same data set in next week. And so hopefully this introduction will help you to get through the homework and to answer the homework questions, but we'll be going in more detail into each of these different steps, particularly the CUF-DIF, the statistics, and we'll be implementing Cummerbund to display the results and take a closer look at individual genes. So with that, thanks for listening, and I'll see you next time.